We live in a world where the possibilities are endless. Biotechnology is booming with new discoveries and even new organisms every day. It's a truly fascinating world to keep up with.
Today, we are going to open the doors to a small room within the big community of biotechnology, genetically modifying embryos, human embryos to be exact. Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a pairing rule. A binds with T and C binds with G.
You might be wondering, what is a genetically modified embryo? An embryo is an unborn offspring in the process of development.
Genetically modifying an organism is done through a process called genetic engineering. Genetic engineering is the direct manipulation of an organism's genome using a set of technologies to take a gene from one organism and insert it into the DNA of another, changing the genetic makeup of the organism. The process to genetically modify an embryo is straightforward but still complex.
There are two basic technical approaches to produce a genetically modified organism: transformation and genome editing. Transformation is the transfer of a gene from one organism into another organism, but we'll be taking a closer look at the technical approach of genome editing.
In the genome editing process, DNA is inserted, replaced, or removed from the genome using restriction enzymes. Restriction enzymes are commonly referred to as molecular scissors. The restriction enzymes are able to cut any targeted position in the genome and introduce modification of the endogenous sequences for the genes that are impossible to specifically target by conventional RNAi.
Conventional RNAi is just a biological process where RNA molecules prevent gene expression, usually by causing the destruction of specific mRNA molecules.
There are currently 4 families of restriction enzymes being used in genome editing today: zinc finger nuclease, transcription activator-like effector nuclease, the CRISPR-Cas9 system, and engineered meganuclease.
The CRISPR-Cas9 technique works like a biological cut and paste tool. Researchers use Cas9 to seek out a particular gene and cut it out of the genome, triggering cell-repairing enzymes to fix breaks. At this time, the target gene can be modified by changing genetic information. The CRISPR-Cas9 technology has already been used to genetically engineer mice embryos. Although the technology has only been around for about 3 years, it has come to be remarkably inexpensive and easier to use in the last few months. Genetically modifying human embryos will be more costly than mice, but still an inexpensive process to perform.
Genetically modifying embryos is a fairly new subject with very little experimentation. There has actually been only one experiment carried out to genetically modify a human embryo to this day, and the information was only released this year.
The researchers used tripronuclear zygotes to further investigate Cas9-mediated gene editing in human cells. In conclusion, they found their experiment highlights the pressing need to further improve the precision of the Cas9 platform.
Since the release of the information, Kathy Niken, a scientist located in the UK, has applied for permission to genetically modify human embryos using a Cas9 technology.
Kathy is a stem cell scientist at the Francis Crick Institute in London and has asked the government's fertility regulator for a license to perform genome editing on human embryos. To help us gather accurate information, we contacted Robin Lavelle Badge, a group leader and head of the Division of Stem Cell Biology and Developmental Genetics, alongside Kathy. Robin has an office located next door to Kathy and was quick to respond with helpful information to support our statements and help us further understand the information.
Kathy still awaits a response from the Human Fertilization and Embryology Authority, also known as the HFEA, since it normally takes about 3 months for a response.
The Human Fertilization and Embryology Act made provisions for the use of genetic manipulation techniques in human embryos for research, but it's also very clear they can't be implanted in a woman to allow further development beyond what is permitted in vitro, which is 14 days.
As long as no technical problems are identified, Mr. Lavelle Badge says they expect that it should take another 2 to 3 months for the license to be granted, as they are optimistic it will be accepted due to nothing in the Act preventing the genome editing techniques they plan to use.
As for many experiments in the community of biotechnology, With this application comes many worldwide questions and concerns. Most concerns are moral and ethical, ranging from arguments about designer babies into environmental impacts and much more.
The reality is there will always be concerns and doubts about new technologies, especially in a community as diverse and broad as biotechnology. It is rapidly expanding in a crucial area for future science. Don't get stuck in the past.
Bill Nye the Science Guy. Bill Nye the Science Guy.